I will give you a brief introduction. So Zena is currently a junior at Amador Valley High School in Pleasanton. She started in ASDRP working in Prabjeet Kaur's group, looking at some sort of microbes in soil. I'm not much of a biologist, so I probably butchered that whole project. But she was looking at microbes in soil. And then she transitioned into management where she thought she was going to be working on chemical biology related. Somebody's making a lot of noise. So what should I do? Oh, I don't know. Please mute yourself if you are not the presenter. Anyways, and so Zena joined my group and started working on a number of synthetic organic chemistry projects, including the one that she's going to talk about today, which is actually the two that she's going to talk about today, one of which targets the main protease of SARS-CoV-2, and the other of which targets the mitotic spindle in cancer cells. So Zena has been using nuclear magnetic resonance spectroscopy to enable synthetic chemistry to develop small molecules to fight human disease. So without further ado, Zena, it's yours.
Thank you for the introduction. So yeah, as Edward said before, my name is Zena. I go to Amador Valley High School. And today I'll just be talking about our studies in the synthesis of SARS-CoV-2 main protease and kinesin inhibitors through the use of computational chemistry or in silico screening with quantitative spectroscopy to make optimized antiviral and anti-cancer agents. So as an introduction or as an outline to what I'll be talking about today, first I'll just introduce our tools that we use, which include benchtop NMR and computational chemistry. I'm going to be talking about the main protein drug targets that we're targeting, and then I'm going to be talking about our actual studies in the in silico screens of Karmifer, the synthesis of Karmifer, the synthesis of Monastrol, and then I'm going to have concluding statements, and also if you guys have any questions, they can be answered. Okay, so as an introduction, a tool that I want to kind of introduce first is computational chemistry, which basically uses simulations to just calculate structures, properties of molecular groups, or reactions, among others, to assist in the process of conducting or analyzing experiments in research. It's capable of doing a lot, such as geometry optimization of compounds through like density functional theory or DFT, homology modeling, molecular docking dynamics, designing compounds for protein binding, and et cetera, that all basically investigate chemical reaction mechanisms, and it's very useful. So what we usually focus on is here in these highlighted boxes, which I'll just walk us through. We use homology modeling or protein modeling to kind of just model our desired protein that we're targeting. And this is shown here in this little kind of ribbon-like structure. And we usually target a protein that is with our compound called a ligand, since it binds to the protein of interest. And we can model the molecular electronic structures of our ligand through density functional theory, also known as DFT, or geometry optimization, to kind of dock our energy minimized ligands to our protein. And this way we can examine their interactions and binding affinities. So sorry about that. I think my computer glitched a little bit with skipping the slides. Another tool that I just want to talk about before I start diving in is NMR, which is a tool that's pretty important to our group, or a nuclear magnetic resonance spectrometer. So based on nuclear structure and what atoms it contains, an NMR can detect on the 19F or fluorine atom, proton, which is 1H, and 13C, or the carbon spectrum. So we use benchtop NMR, which through very recent advances, especially in its proton lock ability, has enabled us to use non-deuterated, or also known as protonated solvents, which are much less expensive than deuterated ones in our experiments, which is great because on a surface level, this means ASDRP can save a bunch of money and we can be greener scientists. But it's also so that we're able to experiment with a lot more options at a much higher scale as well. And through benchtop NMR, it's basically revolutionized the way we can synthesize these compounds, such as through proton NMR, it's possible to elucidate structures, as shown here above, by assigning peaks to parts of a molecule. And this is a podophyllotoxin prodrug that we've been working on. And also through quantitative NMR, we're able to track reaction yields, locate reaction intermediates or impurities, as well as monitor in real-time reaction kinetics, or the amount of time it takes for our reactions to occur. And the ability to track reaction kinetics plays a huge role in our synthetic work. Okay, so the proteins that we're interested in today are the SARS-CoV-2 Mpro and kinesin EG5. The SARS-CoV-2 main protease, or Mpro, is the main nucleophilic protease found in coronaviruses, which is characterized by like a cysteine-histidine catalytic diide in the active site. It can cleave conserved sites of the coronavirus polyprotein, and so in short, this just means that it can inhibit viral replication in cells, which makes it a very important drug target considering its critical role in the processing of polyproteins translated by viral RNA. On the other hand, here on our right, we have kinesin EG5, which is a motor protein essential in the process of mitosis, or cell division. So kinesin proteins are motor proteins, and they move along the microtubule tracts in cells and control the spindle fiber outward orientation and formation. And once inhibited, it can induce apoptosis and reduce cancer cell proliferation. So both of these proteins are essential protein drug targets to controlling disease progression of COVID-19 for Mpro and cancers for kinesin respectively. And the ability to modulate these proteins through small molecule inhibitors that are able to bind to these proteins and control the way they affect metabolic pathways inside an organism, and in this case specifically the way they inhibit their functions, proves to be a very powerful and innovative approach to drug design, which is why we hope to synthesize and optimize 2 of these small molecule drugs. The first one that I'll be talking today is Karmifer that inhibits Mpro. Karmifer is this 5-fluorouracil prodrug and human acid ceramidase inhibitor, and it became a drug commonly used for the treatment of colorectal cancer through its ability to cause ceramide-induced apoptosis. But through very recent drug repurposing efforts, and this is just showing how it's inhibiting acid ceramidase to just create apoptosis. But through recent drug repurposing efforts, we were able to determine that it was also a very potent compound that can bind to Mpro's active site and inhibit Mpro by covalently inhibiting to this catalytic cysteine 145 on the bottom here, whose sulfur, as shown, is attacking this carbonyl carbon of Karmifer and is allowing the release of 5-fluorouracil, which is this head component that is an anti-neoplastic agent that brings major effects to RNA processing and functions. So this makes Karmifer a novel and very promising lead compound for the development of antivirals against COVID-19. But as we've seen with the Delta variant, especially with new mutations in Mpro becoming evident that prevent the efficacy of current vaccines and antivirals, it's very significant to develop drug compounds that maintain the same efficacy with mutants as well, starting the purpose of our mutational analysis studies. So we used homology modeling, as shown in this workflow here, to basically kind of screen analogs of Karmifer, and that could show superior binding affinity in Mpro and its variants. And so we decided to design a library of 33 analogs with varying aliphatic, amino acid, and aromatic fragments against mutations in Mpro. And as seen here, we mainly, again, use homology modeling to just determine any conformational changes in the protein after we mutated Mpro, and tested our ligands to determine which could be hit compounds. So we were able to create a heat map of binding affinities as a result. The red colors indicate compounds with better or more negative binding affinities higher than -8, which meant they were hit compounds. And so we were able to find that the tryptophan side analog, or 2S, had the greatest binding affinity across Karmifer analogs among a variety of Mpro variants, as shown in here, and by increasing the binding affinity above Karmifer in very multiple variants. And so now we were able to just predict how it had great binding affinity, but binding affinity alone does not necessarily indicate that a compound will have very good inhibitory effects against viral replication. And so we continued our studies through actual in vitro laboratory synthesis and testing. So we decided to use isocyanates, which are electrophilic, polar, and extremely reactive compounds in the combination with 5-fluorouracil. And the idea here is that this nucleophilic NH at position 5 here attacks the carbon of the isocyanate, just linking the two to synthesize Karmifer, and hence a variety of its analogs through a bunch of different isocyanate substituents. So here's a more detailed view of the reaction mechanism, which basically we use pyridine as a solvent, isocyanate here, and either through a heat bath or not, we kind of synthesize our Karmifer analogs. And since this compound is extremely polar, we cannot use traditional methods such as thin layer chromatography, since polar compounds, if you know about TLC, they move slower on the plate, and extremely polar compounds such as the compounds we're synthesizing would just not show up at all. So we decided instead to use the fluorine handles on the 5-fluorouracil head to track the reaction through 19F NMR. And so we were able to create these time courses where sodium triflate was our internal standard, as indicated by this like purple highlighted peak. And we were able to make these time courses where green is the product forming. You can see it forming over time going up, and orange is 5-fluorouracil, which is our reactant, going down. And so from there, we were able to make concentration over time graphs, probing the rate of these reactions and optimizing their synthetic reaction conditions to just optimize product formation. And this is major, as NMR had basically enabled us to track a reaction in a quantitative fashion that was seemingly impossible before. So similarly, another molecule we use NMR with, in this case, to probe a reaction mechanism is Monastrol. So this is a dihydropyrimidine privileged scaffold discovered in the Harvard Mitchison lab and is a kinesin EG5 inhibitor. So this image, a very famous image, shows what happens once Monastrol is administered to cells. In a normal cell in figure A, the chromosomes are lining up in the center of the cell during metaphase, but in abnormal cells, as shown in figure C, the chromosomes, which are blue by the way, do not line up properly. And instead, you can see that the microtubules have essentially inverted, so now the chromosome segregation won't be able to happen. And as discovered before, or as covered before, this causes mitotic arrest and apoptosis, which once again makes it a very potent anti-cancer agent. So recently, interest in fluorinated compounds has arose, and not without reason. Fluorine has a very high electronegativity, and it has a greater stability of bonding with carbon, which basically allows fluorination's role in improving drug metabolic stability, bioavailability, and even cell membrane permeability, making it a very popularly used in many medicinal treatments in the industry. And such biologically potent compounds include the ones that are shown all here, which all have a very vast array of purposes. For example, Crestor here fights against cardiovascular disease, fluticasone is an anti-inflammatory drug, and levofloxacin is an antibiotic. Hence, with our ability to utilize 19F NMR, with our ability to utilize 19F NMR, we decided to kind of track the synthesis of fluorinated in this compound. And so as shown here, we have a bunch of compounds, right? And we decided to just fluorinate one of Monastrol's analogs, also LASOM63, shown below, that induces apoptotic cell death in order to just improve both of their antiproliferative and pharmacokinetic properties. So both Monastrol and LASOM63 can be synthesized through the Viginelli cyclocondensation. This is a 3-component reaction between 3 reactants, an aldehyde, beta-ketoester, and urea. So multi-component reactions are interesting as they offer an opportunity to basically study various reaction mechanisms, as oftentimes they can proceed through a lot of them. For example, the Viginelli can actually proceed through 3 different mechanisms that all produce the exact same product, including the imine, enamine, and novinagel. The preferred mechanism through which this reaction proceeds has been greatly debated, and some debate the existence of the 3rd route that I named the novinagel mechanism due to its thermodynamic unfavorability. And so even though previous studies had been creating analogs by creating ester substitutions at this R1 position as shown in blue, we introduce a new question of the substitution of a CF3 group and its mechanism or effects on the Viginelli in the C6 position instead, as shown by this red part in R2, which is not studied as thoroughly. So by using 19F-NMR, we monitored the synthesis of trifluorinated analogs of Monastrol and LASOM63. And in both cases, we realized that this final dehydration step didn't occur properly. And so if you see below on our final compound with the trifluorinated group, this OH is there, which is not what we want to see. And we discovered this was because the CF3 group is actually preventing the dehydration from happening. And so through additional COSY spectra, we also saw this coupling between a benzylic proton and the neighboring proton, which once again just kind of proved that this final dehydration cannot occur. But through kind of using another reaction where we kind of used tosic acid conditions instead, we were able to carry out a final dehydration, just not in one pot, yielding our 6-trifluoromonastrol and 6-trifluoro-LASOM63. But we also observed an additional 19F peak while monitoring the synthesis of our trifluorinated LASOM63. And this unexpected peak that showed up, we had no idea what it could be. And so our group members, we kind of had a hypothesis that it belonged to a possible reactive intermediate. So we synthesized the intermediate and checked its 19F chemical shift, and it turned out that it was actually a match. So we determined that this additional signal belonged to a Nova-Nagel adduct between the aromatic aldehyde and the beta keto ester, whose fluorine residence you can see is kind of in purple here. And this suggests a change in the reaction mechanism because in contrast, Monastrol and its trifluorinated analog are forming through the condensation of an aldehyde and a thiourea instead, which was very interesting because, as discussed before, the Viginelli reaction rarely follows this reaction mechanism, which we call the Nova-Nagel route. I've talked about this previously because a lot of computational and mass spec studies have both repeatedly demonstrated that this mechanism poses a great thermodynamic barrier, hence being very thermodynamically unfavored. In fact, some people have even argued that this reaction doesn't exist because they believe it's simply not possible. But we were able to prove otherwise with our mechanistic studies with 19F-NMR that yes, it does proceed. We also isolated a product of this reaction, shown below, the Hansch reaction, which is very similar to the Nova-Nagel, and this product probably formed due to the occurrence of Nova-Nagel route, kind of showing how we were able to use 19F-NMR to override previous predictions made by computations and kind of probe reaction mechanisms, which is an extremely interesting concept to kind of optimize the molecules and the drug compounds that we're synthesizing. So in conclusion, I've talked about how we are trying to synthesize and create better antiviral or Karmifer and anti-cancer or Monastrol derivatives. And in such case, with Karmifer, I discussed how it inhibited MPRO and how we can further use our isocyanate substituted analogs to test our ability to inhibit MPRO and see how well it happens. And same with Monastrol. We are very interested in further studying if it's able to be a more potent compound. So for future directions for Karmifer, we're going to continue synthesizing our isocyanate substituted analogs, and we're also looking forward to biologically testing them on actual MPRO proteins, which will be very interesting. And for Monastrol, we also want to kind of hone in on that biological testing part to kind of do cell line, cancer cell line assays. And also because we were all able to probe the mechanism of the Viginelli, we also want to do a further study of the other Viginelli routes, either through aromatic substitutions or through the hopes that we can actually study another possible mechanism, the Pechmann condensation. So in conclusion, I just want to give some acknowledgements. One, to my advisor, thank you very much, Mr. New, and as well as our donors who have generously made it possible for our research to happen. I also, although not listed on this slide, want to give a tremendous thank you to all of my coworkers who are such a joy to work with. They've guided me through a lot of my path through research, and they've done all very incredible work. So thank you for listening. Are there any questions?
Sure. You made some comments about the Novonagal route being difficult because of the high thermodynamic barrier. How does that manifest itself, and does this mean a smaller yield? How did you work through this?
So we actually kind of stumbled upon this route kind of, I guess, in like a lucky kind of circumstance. We didn't actually try to pursue this route, if that's what you're asking. We stumbled upon it while we were trying to create one of our trifluorinated analogs, and we were able to kind of see how it's interesting that this route is able to actually proceed.
Okay. On the Karmifer, how many analogs have you made?
So far, we've just been making, I can kind of go back a little bit. So far, we've just been making the trifluorinated versions of it, which is essentially just attaching these CF3 groups to Monastrol and a CF3 group to LASOM63.
And have you tested those yet?
We are in the process of testing them.
So that's the next.
Yeah, that's one of our.
Okay.
Yeah, so that's one of our future directions.
Okay, I think that's all of my questions. You have something on chat.
Oh, okay. Let me check it. Oh, can you go into a little bit more detail about the specificity of the drugs and how they target pathogenic or cancerous cells? Yeah, so Monastrol, or I can kind of first talk about Karmifer a little bit. I'm going in the opposite direction. Karmifer, as shown here. Oh my God, okay. Karmifer, as shown here, it's basically able to kind of target this sulfur on the cysteine 145, which is a catalytic diad, and basically it is able to attack it, and once attacked and binded to, it's able to release 5-fluorouracil, which is how Karmifer kind of specifically binds to MPRO and is able to release our 5-fluorouracil. And for Monastrol, in this case, it's a dihydropyrimidine, and it's able to kind of bind to kinesin instead. And it's very interesting because in the way that it inhibits it, it basically just doesn't let the motor protein conduct its usual pathway, which is just to actually properly form the spindle fibers, which is kind of the reason why that it can actually cause apoptosis and also cell death. So that's how it kind of targets cancerous cells, right? Because cancerous cells are continuously dividing. And basically the way that it kind of stops cell division or kind of slows it down, it's basically targeting those cells that are dividing so quickly.
So I want to jump in again. I want to say that you are very clear. You do a very good job of explaining things that you don't hesitate. You have all of the statements that you need to make in order to explain it so that somebody who had any idea of this could understand it. There is one small change in speaking style that I might suggest, and that's lose the kind of. When you're explaining a process, you says, this process kind of goes this way. Just say this process goes this way.
Yeah, that is a kind of speaking habit I'm trying to correct a little bit. Thank you for telling me.
A kind of speaking habit?
See, I did it again.
It's going to be a long. Okay.
Super job. Really super
great job. Great job.
Yes.
Yes,
Thank you very much.
I want to jump in and say that, yes, that's a wonderful thing. So sorry if I sound like a critics, but it is not critics. We are here to improve our researchers, skills from all the angles. So, you know what, as a person who is listening to your presentation, I'm like, when I meet my researchers, that's the first thing that I emphasize. When we have so many slides, and when people, they have questions, they want to go back to a slide, to a specific slide. It's hard now that I want to tell you what questions I have because I don't have a track of the slide number. Make sense? So I'm lost now if I want to ask anything, like which slide it was. Like if there was a slide number, then I would have not done, okay. So, you know, jot down that, okay, you know, I'm looking for slide number 12. Can you go back and tell me what you mean on slide number 12? Especially when we have a lot of slides, please make sure that you have, just for all, not you, but all the people who are in this colloquia, having a slide number has a meaning. Sometimes when you are presenting in big audience, like, okay, today I'm going to walk you around, like I have made around 13, 14 slides. So just also when you are on slide number 2 and you have slide 13 slides, so people are like, oh, she's gonna talk a lot, you know? So it's basically giving some idea to the audience also that how much of their time you're going to take. So there's a lot of hidden value behind that slide number. So that's one thing. Second thing, from a person who is coming from bioinformatics, when you started explaining about computational chemistry. So basically that is not on your second slide. That's not at all computational chemistry. So when you talk about those helix of RNA and DNA, that's actually computational biology and has nothing to do with computational chemistry. Computational chemistry come to picture as chemoinformatics when we are talking about the drug discovery, the ligands, the binding, the docking. So that becomes, they call it computational chemistry, but as in it's known for the topic as the drug discovery or chemoinformatics, just to make it like clear about like those arrows that you mentioned and explained about computational chemistry. So in one of the slides, I don't know which one it is, when you showed NMR, there was a formula of a cos square of something plus cos of something. So do you know where that formula is coming, that equation is coming from? Are you aware that what is that equation is about?
It is a very specific equation, but currently it's just not coming to mind the name. I'm kind of not remembering the specific name. Okay. But it's just basically describing product formation, how we aren't able to predict.
Maybe if you can walk to that slide, I don't know what's the slide number, so your friends can see what I'm talking about, maybe a little bit further.
Okay, sure.
Yeah, there it is. So yes, that equation. So do you know how it is relevant? I mean, you answer me no, but do you know, like in any spectra that you get, how that equation changes? That's pretty interesting, actually. So I will leave it if you have not come across. So just leave it. I was curious that maybe if you have done some mathematical modeling on that, then you have your own equation, because I saw the sample equation on that.
So I'll actually answer that, because I think that equation came into the story prior to Zena joining this project. We were interested in the stereochemistry of the two methine protons and the chemical structure that's drawn right below the equation, and that's the Karplus equation. So it relates a particular coupling constant, which you can extract out of the spectroscopic data, with the dihedral angle between two protons. And so we did a little bit of quantum mechanical modeling to identify what possible stereoisomers those two protons might be in. So that's why that equation has made it onto every single figure ever since. It's a good question. I think for all of you researchers out there, I think it's an important thing. If you have an equation on your slide, be prepared to talk about it and what it means. And not only what it means, but why it's important for your science.
I have no further question. Good job.
Okay. I just think I have one more question, which is, do the mutations in the binding site of the Delta variant and other variants change the binding affinity of your drugs? Yes, it does, because once again, different mutations in the binding site, they have different kind of properties with the binding of your substrate. As you've probably learned in biology before the lock and key model, there's usually specific substrates that can bind to specific subsites. But our hope is that we can create a kind of specific enough, but also in the same case general enough potent inhibitor that even if there's a variant, because the substrates are so similar to the wild type of MPRO, it'll also be able to do pretty significant inhibition. Okay, if there are any questions, that's the end of my presentation.
Great job, Sina. I think we have Robert. Do you want to introduce your speaker for the day?
Sure. Rahul is, pardon me, part of the Voynich manuscript group, has been for a while now, and I would like to say exemplifies the true nature of research in taking what is a pretty standard encryption, decryption, natural language processing approach to decoding the manuscript, and went a completely different direction. And I'll let him explain the significance of it, but he started looking at the physical properties of the manuscript as opposed to the reputed content, and has really found some strong ties that may shed light over time as we continue doing the research on the original source language and the intent of this manuscript. So with that, Rahul, without having taken too much of your thunder, I hope, I'll let you go ahead.
Hello, everybody. My name is Rahul, and today I'm going to be talking about the Voynich manuscript and its potential origin in Persia. So what exactly is the Voynich manuscript? Before we begin, I would like to explain some definitions. First, a folio. A folio is basically a parchment leaf, which is basically ancient paper. The second definition is called a quire. A quire is a collection of leaves, which is basically similar to a chapter in a modern day book. The Voynich manuscript is a mysterious manuscript which was made in the early 15th century, dated around 1430. It consists of 102 folios. Originally, it had 116, but 14 are presumed to be missing. It used to have 20 quires, but currently there are quires 16 and 18 missing. It was named the Voynich manuscript due to it being brought to light by an antiquator named Wilfred Voynich in 1912. It resides in the Beinecke Library at Yale University. When Voynich first got it, he held onto it for a couple of years and referred to it as the ugly duckling of his collection. When he died, his wife inherited it, and when she died, his secretary inherited it. Later, antiquarian H.P. Cross bought it for $24,500 and tried selling it for over $160,000, but unfortunately, he didn't find any buyers for it. In the end, he donated it to the Beinecke Rare Book and Manuscript Library of Yale, where it still resides today. The Voynich manuscript is written in a mysterious language which cryptologists have tried and failed to crack for hundreds of years. Notable attempts include the Jesuit Athanasius Kircher in Rome in the 17th century, and William F. Friedman and his wife Elizabeth Smith Friedman. It contains 6 different sections. The sections include a herbal section, astronomical section, cosmological section, biological section, pharmaceutical section, and a recipe section. The herbal section includes drawings of herbs, some real and some which may be imaginary. The astronomical section includes drawings of the sun, moon, stars, and what are thought to be zodiac signs. The cosmological section is mainly just circular drawings. The biological section has anatomical drawings of people in tubes of yet still unknown liquids. The pharmaceutical section, which basically has containers next to small herbs and stars next to them. And finally, the recipe section. The recipe section has over 300 paragraphs of still yet undeciphered recipes with stars next to them. For a little fun tidbit, the Voynich manuscript's official name is MS 408. Now, what does the Voynich manuscript say? As I've stated before, the manuscript is still undeciphered, though many researchers have claimed that they've deciphered some words using hieroglyphs. There are many assumptions on what it could mean, ranging from early inventions to random scribbles to even a rare prayer book. The field is a field comprised of academic, governmental study, and amateur study, so it is by nature unbounded. Fun fact, many people claim to have cracked the manuscript, but as there isn't enough evidence supporting them, all of the claims have been dismissed. There are at least 3 claims of the origins of the manuscript being found or the manuscript being cracked. given every week. Now for the current work going on in the field. Different teams of researchers are trying to use different methods to try and decrypt what the manuscripts could mean. One group of researchers is using a method called algorithmic decipherment in order to decrypt the manuscript. Algorithmic decipherment works by enciphering text by converting it into ciphers of the same length and then deciphering it from there. Another group of researchers is using a method called anagram decryption instead of the other two methods, or are using the other two methods in conjunction. Anagram decryption works somewhat similarly by scrambling the text and then decoding it from there. What we are doing here at ASDRP. So the origin of the manuscript has not still yet been identified. Instead of initially focusing on a manuscript whose language is still indecipherable to us, we decided to take a look at something that would be more tangible and hopefully a little more easy to accomplish. We are looking at tracing back the manuscript to where it was originally written. If the original place is found, researchers can compare the language to other written languages in the same area, in the general geographic area at that time, so we can decode the manuscript by comparing it to other languages that were there. Of course, it's simply a possibility, but the chance of all of the constituents of the inks to be widely used or found in Persia is too slim to merely be a coincidence. Our findings. We found similarities to Persian characters from the letters in the manuscript through character mapping and combining both Cower and Knight's method to find the character frequencies of each character in Voynich. After finding the similarities, we decided to trace back the constituents of the manuscript back to Persia by researching whether the materials used to make the manuscripts were available in Persia at the time, as well as researching if manuscript writing was prominent at the time. Now here are the inks used in the manuscript. The black ink which was used to write the text is known as iron gall ink. As you can see in this image right here, that is an excerpt from the manuscript which shows the iron gall ink being used, as well as some other dyes such as green and a little bit of red. The blue ink used is ground azurite, which is shown right here. The green ink is copper chlorine resonate, also known as verdigris. The Statue of Liberty's current color is actually known as copper green or verdigris. The red ink is known as red ochre, also known as hematite, which is right here. And the white ink is known as calcium carbonate, which is depicted right here. Now I will explain the ink's direct links back to Persia. Iron gall ink. The ink used to write the majority of the words in the manuscript was identified to be iron gall ink. Unlike typical iron gall inks, however, this ink possessed some abnormalities in the composition, mainly containing things such as zinc and copper, which were thought to be from an inkwell. At the time of the manuscript's writing, brass inkwells were commonly used in Persia, which contained both zinc and copper. While many other countries had individual mines of zinc, copper, and zinc, for instance, Germany had a massive mine of copper around the time, Persia had massive stores of both and used to export them to other countries as well. A source known as the Kokuyao claims that Persia was the first country to mine both elements and use them to create brass. The Kokuyao originate from China, which could be a potential connection to the Silk Road, which will be discussed in a later slide and why it is significant. The blue ink used in the manuscript was called ground azurite. The term azurite comes from the Persian term lajwad, I'm sorry if I'm butchering the pronunciation, which means deep blue. During the medieval ages, the recipe for an ink called Egyptian blue, which was widely used around the time, was lost. So the only source of blue around the time was azurite and ultramarine, which was from Afghanistan, which was a part of Persia at the time. If a term originates from a language, it usually implies that the country has or has had access to the material in the past. This makes it easy for Persia to have access to azurite as an ink. After that is the green ink. The green ink in the manuscript was identified as a copper chlorine resonate. It was a very popular pigment in use for many paintings during the medieval ages. It was the main green used among Persian manuscripts, with the majority of the manuscripts that were studied. Out of 45 manuscripts studied, 22 of them used copper green or verdigris as their main green. Other elements that were used in the green ink included azacamite, which is also very common in Persian manuscript used at the time. While it was not as common as verdigris, it was still a usual component of making verdigris. After that is red ink. The red ink used in the manuscript was identified as red ochre, also known as hematite. As with green ink, the primary red ink used in Persian manuscripts were identified to be hematite, with not only 22, but 26 out of the 45 of the manuscripts being studied having used hematite as their primary red ink. The Persians also had a regular source of hematite from the Hormuz Isle, which is pretty close by to the mainland, making it easy for the Persians to export it from the Hormuz Isle, exporting it outside to other countries, as well as using it as in their own manuscripts. Finally is the white ink. The white ink used in the manuscript was identified to be calcium carbonate, also known as egg layer. Compared to the other inks, though, this ink is an oddity. While the other inks have multiple records of use in Persian manuscript, this ink is far less mentioned, as white lead was a primary white ink used in Persia. There are some occurrences of this ink being used, but they are far fewer than the other ones being used in manuscripts. In fact, I would say that this is, if in those 45 manuscripts, I would say that only about one of them used calcium carbonate as their main white ink, while the other ones mostly used white lead. Additional historical ties. The Silk Road. During the period the Voynich manuscript is dated to have been created, which is around 1430, the Silk Road, which is an ancient trade route, weaved throughout the Persian Empire as well as many other empires, playing an important role in the exchange of both goods and thoughts. The route transported silk, porcelain, paper, spices, and most important of all of the inks described in slides 10 to 14. This is extremely significant because it contextualizes the time period in which the Voynich manuscript is dated to have been written, and it provides a clear link to the Persian Empire in terms of access to the inks necessary to construct the manuscript. Additionally, the fact that the manuscript contains rich information on herbology, medicine, astrology, and more indicates that the Voynich manuscript authors were likely influenced by the vibrant exchange of knowledge occurring through the Silk Road. My personal theory is that it was probably a traveling merchant who was traveling through the Silk Road and was in Persia at the time that he or she wrote the manuscript. The reason I think so is that it's not a noble is because nobles and kings around the time the manuscripts were written, they always used gold leaf as gold ink in their manuscripts, which is mostly exclusive to upper class, higher, wealthier people. So I think it was probably just a regular merchant traveling through, but he learned many things throughout the Silk Road and decided to jot them down in the Voynich manuscript. And also, during the time of the manuscript's creation, Persia received a massive increase in manuscript writing, many of which are viewable in the Met Museum today. For future research at the ASDRP, as the research for the summer semester was mainly concentrated only on one country, which was Persia, future research will be expanded to track down more of the inks locations throughout the Silk Road, so we can find out how many inks were in each different country around the time, so we have a more broad pool of languages to look at. We also intend to broaden our historical tracing efforts and use character frequency analysis to compare the frequencies of letters in Persian and Voynichese. We will branch out to involve native speakers of Persian to also take a look at the manuscript and identify other similarities which we have missed. We have already begun this process, however, they are a little bit too busy right now, but we will incorporate this into our later findings. For acknowledgements, thank you to Mr. Downing for helping us to get started on research. Thank you to my colleague Aylin for bringing up the idea that the manuscript may have been linked to the Persian Empire based on character mapping analyses. And thank you to ASDRP and all of the donors for allowing us to conduct research here. Are there any questions?
I may be biased, but great job.
Thank you, Mr. Downing.
I have some questions.
Yes?
Have you cooked any of the recipes?
Unfortunately, I have not.
Have you actually deciphered any of them?
We haven't exactly deciphered any of them, but however, we have found some similarities to Persian characters. And as of now, one group claims to have cracked at least 9 words out of like hundreds of thousands of words in the manuscript, including words such as I think one word meant cow.
You said that you used the presence of the inks in other Persian manuscripts to indicate that that was a likelihood of it being done in Persia. Given what you said in your talk, why couldn't it have been done anywhere on the Silk Road?
Because as I said in one of my earlier slides, there are many inks I agree with that, and that it could be anywhere on the Silk Road. But the fact that all of the potential inks and their usage in manuscripts were concentrated in one particular country, which in this case is Persia, and that the country had easy access to almost every single ink, especially to the fact that calcium carbonate is just egg there, which is easily makeable. I have my theory is that it was most likely originated in Persia, but we don't know that for certain, which is why for future research, we are going to be branching out and looking at like other places where the inks could have originated from.
Okay. And the other thing you mentioned, which I thought was very interesting, was that noblemen, if they're going to use a gold color, they use gold leaf. But apparently this was not a nobleman, he was a businessman. So what did he use for a gold color?
The interesting thing about that is that there was actually no gold color discovered in the manuscript. All the inks that I listed were the only inks that were used in the manuscript. So surprisingly, there wasn't any gold leaf used in it at all.
So was there any yellow?
I would say it was yellow. I would say maybe it was like red that eventually faded with time. But there was a little bit of a proteinaceous substance on the surface of the pages, most likely used to preserve the pages over time.
All right, great. Thank you very much. It was very fascinating.
Thank you too.
Hey, Rahul, I go second. So I might be able to help you with this project because I'm Persian and I speak Farsi. I mean, I speak Persian. So yes. Speaking of this, I know that right back, so now Persia has shrunk and the only country who is people speaking Persian is Iran. But I know in universities in Iran, like back then when a couple of my friends were studying, they do decoding on this one and they believe that this is coming from Persia. I'm reading my DMs. Okay. Yes. So they believe, and I think they made some progress, but again, it is going back to getting translated to the native Persian language. And it is, for some reason, of course, they are not sharing it with the world. I don't know what's the reason, but it is like the fact that maybe they're trying to decode it from English perspective, it's new. But looking at this, it is not something new back at home. Like I know there's so many people who they study anthropologists and linguistic in Iran. That's like part of their course and research ideas. Like they're still going on, I think for years. I don't know what they're doing. But yeah, they're still trying to decode this. And just a couple of things, like when we're talking about Persia country, it was not a country, it was like a small country. So I would say, let's use the word Persian Empire. It will be curious. I mean, it will be nice if next time you show the geographic map of Persian Empire, that it was from Egypt back till India and north. And so basically we're talking about like 1/3 of the globe being Persia. So it's no wonder if we are trying to, decode it and, finding all this pieces together. But great job. I'm very impressed and curious. So please, if you need anything and you think I'll be of help, I'm absolutely have time for you to answer your questions.
Thank you for the feedback, and we will definitely reach out to you if we ever need help. I really, really appreciate the offer. Thank you very much.
Anytime.
Yep, she's on the hook now.
Following up on Sahar's comment, she said that you should have a map of Persia there. It would be really nice if you drew a picture of the route of the Silk Road on that map.
All right,
so that people could see where the movement back and forth is being done.
I'll definitely make that, and then I'll share it with everybody. And I think there's a question in the chat. Has research found reasons as to why different ink was used in these manuscripts? Did they have a specific purpose or use, or are they used specifically to determine the origin of the manuscripts? So as for the different inks, I say that the reason different inks, I'm assuming you're referring to the colors. I say that the different inks were used because while the main primary ink for writing it was just like iron gall ink, which was a little bit of a blackish color, the other inks were used to basically illustrate the things, like create drawings of things, like such as different plants and different people. And they did probably have a specific use for using that to demonstrate how the plants and stuff looked. But for the research, we are simply using it to determine the origin of the manuscript. My colleague Aylin, she speaks a little bit of Persian, and that's how we initially found out that the manuscript could initially like actually be from Persia. Are there any other questions? One second. Let me see. Did eggs originate from Southeast Asia? How come Persia is the original source of the white color coming from bicarbonate? That's the thing. Like during my slide about white, it's actually not. It didn't come from that original area. Persia did have access to it, but it wasn't their main ink. Persia wasn't the original source of it. Usually Persia for almost every single manuscript that I studied, almost every single ink used there was white lead. The calcium carbonate was a little bit like, it was an abnormality. Not all countries use that. Like countries that were in more of Europe, the Europe area, those countries used to use bicarbonate, egg glare, things like that, calcium carbonate. But for Persia, they mostly used white lead, which was why I was surprised to find out that this one was actually calcium carbonate as well.
That might be a clue to the origin of the person who did the manuscript. Maybe he brought the white carbonate ink with him.
Or perhaps he may have imported it through the Silk Road.
Have imported it because that was what he was familiar with.
Mm-hmm. Are there any other questions? I guess not. Thank you so much for your time, everybody. I hope you all find it as intriguing as I did, and I hope you all have a nice rest of the day. That's all for my presentation.
Thanks, Rahul. Again, great job.
Thanks, Mr. Darning.
Agreed. Well done.
Edward, is there an attendance thing today?
Don't forget to fill out the attendance form, which is in the chat window. Here it is the third time. That's how we know that you are here.